Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Pondicherry University MSc Entrance Exam question paper for the subject Biochemistry and Molecular Biology, which was conducted in the year 2017. I will be discussing only the biology part. So coming to the first question. In enzyme kinetics, Vmax reflects. So Vmax is the reaction rate when the enzyme is fully saturated by the substrate, and it indicates that all binding sites are being constantly reoccupied. So answer is enzyme substrate complex. Option alt 1. Alternative 1. So what is true about gap junction? Gap junction proteins are composed of 6 connexin subunits. So made up of connexin proteins is the correct answer for which is true about gap junctions. Then production of excessive amount of corticotropin ACTH occurs during the Cushing's syndrome. Answer is option D. Then next one. Which one of the following interactions play a major role in stabilizing the B form of DNA? So the hydrogen bond, it plays a major role in stabilizing the B form of DNA. Then megaloblastic anemia is caused by the deficiency of. So it is caused by deoxyadenosylcobalamin. The active form of cobalamin, that is vitamin B12, is deoxyadenosylcobalamin. So megablastic anemia is caused due to deficiency of deoxyadenosylcobalamin. The phenomenon of interchange of functions between related genes are called. So answer is complementation. So what is genetic redundancy? It is a term used to describe situation where a given biochemical function is redundantly encoded by 2 or more genes. Complementation, it refers to a relationship between two different strains of an organism which both have homozygous recessive mutations that produce the same phenotype but which do not reside on the same homologous gene. So the phenomenon of interchange of function between related genes is called as complementation. So there are two organisms which have mutations or homozygous recessive mutation that produce for the coding for the same phenotype. So and they do not reside on the same gene. So one if the gene A, if there are two genes, that is A and B, if A is mutated in one and B is mutated in the other, the one which is having the non-mutated form of A will code for protein A, and one which is having the non-mutated form of B will code for the protein B, and they both will function together. So it will look like there is no mutation phenotypically. The phenomenon of interchange of functions between related genes are called complementation. Then genetic interaction is the set of functional association between genes. So answer is option B, that is complementation. Then the most commonly used molecular tool for phylogenetic analysis involves sequencing of. So it involves sequence of ribosomal RNA. For phylogenetic analysis, 16S rRNA is used, especially in case of bacteria. Then Mullerian inhibiting substance. So inhibits Mullerian duct differentiation. So androgens secreted by lady cells induce differentiation of Wolfine ducts into male internal and external genitalia. So Mullerian inhibiting substances, it inhibits Mullerian duct differentiation. Then next one. A ribozyme is? It is a class of RNA molecule that can catalyze chemical reaction. So that is a ribozyme. The distinct foci within the cytoplasm of the eukaryotic cell involved in mRNA turnover is called. They are called as P bodies or processing bodies. Answer is option D. The pollutant released by jet planes is aerosols. This is a repeated question. Oncogenes are cancer-causing genes in the cell, but they do not express usually. This is because there is what is called as the tumor suppressor gene. That is why oncogenes, even though they are cancer-causing genes in the cell, they do not express because of tumor suppressor genes. Then acrosome of a sperm cell is modified. It is modified Golgi. Then synthesis of glucose from amino acids is termed as gluconeogenesis. Option B. Which of the following statement concerning glucose metabolism is correct? Glucose enters most cells by a mechanism where Na plus and glucose are co-transported. The conversion of glucose to lactate occurs only in RBC. Not only in RBC, it occurs in muscle cells also. Pyruvate kinases catalyzes an irreversible reaction. Now that's correct if you see the various steps of glycolysis. So alternative 3 is correct. Elevated levels of insulin reduces level of fructose 2,6 biphosphate in hepatocyte. That is also wrong. So the correct statement concerning glucose metabolism is pyruvate kinase catalyzes an irreversible reaction. Then choose the amino acid having maximum number of codons. So alanine is having 4, leucine is having 6, tryptophan is having 1 and valine is having 4. So the one which is having maximum codon is leucine, that is 6 codons are there. The genomic DNA sequences similar to normal genes but non-functional are called. So they are called as pseudogenes. Option 3. The unfolding of regular secondary structures causes a large increase in entropy of the protein. So answer is option 2. That is large increase in entropy of the protein. Then next one. Golden rice is rich in carotene and iron. Answer is option C. Lipopolysaccharide in gram-negative bacteria is found on the outer membrane. Then next one. Blood group type A antigen is a complex oligosaccharide which differs from H antigen present in type O individuals by the presence of a terminal N-acetyl galactosamine. So blood group type A antigen differs from H antigen present in O type individual because of the presence of terminal N-acetyl galactosamine. Then next one. As one proceeds with the purification of an enzyme with every subsequent step, the enzyme activity. So total activity is the total number of enzyme activity units recovered at each step and present in the sample from that step. It will usually decrease during purification. So total activity will decrease during enzyme purification. Why? Because protein is lost in various steps and protein is also denatured during manipulations. That is, manipulations means during various purification procedures. But purity of the enzyme increases. So even though total activity decreases, the purity of the enzyme decreases as you go through various purification steps, and hence specific activity increases, whereas total activity it decreases. So answer is option A, decreases. The study of cancer is referred to as oncology. Option 4. Then monooxygenase important for the detoxification of many drugs. It is a cytochrome P450. Then next one. On the molar scale, which of the following interactions in a non-polar environment provides highest contribution to biomolecules? That is hydrophobic interaction. Then Mycobacterium is an intracellular parasite. It prefers to infect macrophages. Then next one. The amount of disorder in a system can be expressed as, it is expressed as entropy. Then glycogenin is, it is a protein primer for glycogen synthesis. Just like you have a primer for the synthesis of the DNA, there is a protein primer called glycogenin for glycogen synthesis. Which of the following amino acid does not have optical isomers? So glycine doesn't have optical isomers. Then cataract is a disease caused by, it is caused by a clouding or loss of transparency of the eye lens due to tissue breakdown and protein clumping. So cataract is a disease of the eye. It is caused by caused by clouding or loss of transparency of the eye lens, and it is due to tissue breakdown and protein clumping. Then marasmus is characterized by, so marasmus is characterized by severe protein and calorie deficit. Then next one, a pair of genes controlling a pair of contrasting characters is called as an allele. So answer is option 4. Then next one, the allowed regions in the Ramachandran plot for 3 residues alanine, glycine, proline decreases in the order. So glycine greater than alanine greater than proline. This you have pictures either in the internet or in your biochemistry books. So you can refer that. Then number of base pairs per complete turn in zDNA. It is 12. For bDNA it is 10. For zDNA it is 12. HMP shunt is unique in generating 2 important products. That is, the HMP shunt always plays a significant role in NADPH2 formation and in pentose sugars that are biosynthetic precursors of nucleic acids and amino acids. So it generates 2 important products, that is pentoses and NADPH, not NADH. Then next one, the microorganism that is mainly used as an indicator of fecal pollution in water is Escherichia coli. This is a repeated question. Then next, AIDS is not transmitted by, so AIDS is not transmitted by mosquito bite. Option A itself is correct. So through mosquito bite, AIDS is not transmitted, but sharing unsterilized needles, unprotected sex and transfusion of infected blood, AIDS is transmitted. An anti-hemorrhagic agent is a substance that promotes hematostasis or stops bleeding. Which of the following vitamin can be considered as an agent? So they have asked for the one which stops bleeding. It's an anti-hemorrhagic agent, and which of the following vitamins that is considered as an agent for to stop bleeding? So answer is vitamin K. The plant hormone auxin causes cell expansion. This is a repeated question. They have asked for 2-3 years. Then ethanol decreases gluconeogenesis by. Now ethanol, it inhibits hepatic gluconeogenesis by decreasing the steady state concentration of pyruvate as a result of decreased NAD plus and NADH ratio. So converting NAD plus into NADH and decreasing the availability of pyruvate, that is option 2 is the answer. That is how ethanol decreases gluconeogenesis. The next one, cyclic AMP is formed from ATP by the enzyme adenylate cyclase which is activated by the hormone. So this is activated by the hormone epinephrine. Most common type of phospholipids in cell membrane of nerve cells. So sphingomyelin is a type of sphingolipid found in animal cell membrane, especially in membranous myelin sheath that surround some nerve cells. So answer is option D, that is sphingomyelin. Then next one. Sucrose consists of glucose plus fructose. Then fatty acid biosynthesis requires dash for the transport of acetyl-CoA from mitochondria. So fatty acid biosynthesis, it occurs in the cytosol. So acetyl-CoA has to be transported to the cytosol from mitochondria. Now this acetyl-CoA, it cannot cross the membrane. It is transported out of mitochondria as citrate. Citrate is formed by condensation of acetyl-CoA with oxaloacetate by the enzyme citrate synthase. Then after reaching cytosol, citrate is converted back to oxaloacetate and acetyl-CoA is released. So that is how acetyl-CoA is transported from mitochondria to the cytosol during fatty acid biosynthesis. So answer is citrate, option 3. The chemical typically released by the body in an allergic responses, they are histamines. Then mosquito act as a vector for the disorder. So Bancroftian filariasis. For that, mosquito is the vector, whereas for others mosquito is not a vector for other disease transmission which are given in the options. The technique for purification of proteins that can be made specific for a given protein is. So if you want to specifically purify a particular protein of interest, then affinity chromatography is the best one, because that is based on the principle of binding of a particular protein to its ligand. That will be highly specific. That type of reaction, that is ligand to protein reaction, it will be more highly specific, and only a particular protein will be able to bind to the ligand during purification. So the technique for purification of protein that can be made specific, so specific I have asked, for a given protein is affinity chromatography. Carcinomas are tumors arising from, they are tumors arising from epithelial tissues. Then next one. Bacteria protect themselves from viruses that infect them by fragmenting viral DNA with the help of. So bacteria, they protect themselves from viral infection by using restriction endonucleases. That will, what will it do? That will fragment viral DNA which enters into the bacterial cell. Sorry. So answer is restriction endonucleases. Liquid food drinking is, it is called as pinocytosis. Then all of the following statements about the enzymic complex that carries out the synthesis of ATP during oxidative phosphorylation are correct, except. So it is inhibited by oligomycin, that is correct. It is located on the matrix side of the inner mitochondrial membrane, that is also correct. It can exhibit ATPase activity, that is also correct. It can bind molecular oxygen, that is not correct. So the correct answer is it can bind molecular oxygen. The next one. Mammalian promoter sequence is located. So it is located at about 20 base pair upstream of the transcriptional start site. So where is the promoter sequence located? It is located upstream of the transcriptional, not translational. It is transcriptional start site. Then nitrification is the conversion of. So nitrification is the process that converts ammonia to nitrite and then to nitrate. So ammonia is NH4 plus and nitrate is NO3 minus. So answer is option 4. The next one. Phosphatidylserine is an important component of biological membrane is located in So it is located in the inner leaflet but flip-flop to outer leaflet under specific condition. So phosphatidylserine is located on the inner leaflet but it flip-flops to outer leaflet under specific condition. So what is it means is phosphatidylserine, it is an anionic phospholipid that is usually localized in the inner leaflets of plasma membrane. But the enzyme scramblase, it catalyzes the externalization of phosphatidylserine on the outer leaflet of the plasma membrane during apoptosis or cellular stress condition. Next one. Which one of the following would be expected in pyruvate kinase deficiency? So if you can expect hemolytic anemia. Answer is option B. Then next one. In fluid mosaic model, so what is fluid mosaic model? The proteins are embedded at places in phospholipid bilayer. All the other options are not correct. One of the following statements is correct. Insulin converts glycogen synthase B to A. Now that is correct. So when option A itself is correct and only one of the option is correct out of 4, answer A is the right answer. So glycogen synthase A is the phosphorylated UDP glucose molecule interact and grow into a glycogen tree. CAMP converts glycogen synthase B to A. All these are wrong and only option A is correct. So coming to the next question. The size of red blood cells in venous blood is greater than that of arterial blood. This increased size of red blood cells in the venous blood is due to. Now the difference is due to the exchange of carbon dioxide for oxygen as the red blood cells moves from artery to vein. So what happens is carbonic anhydrase converts the carbon dioxide to H2CO3 which spontaneously dissociates to H plus and HCO3 minus ions. Now there is a transport protein that exchanges this HCO3 minus for Cl minus. Now what will result is it causes osmotic pressure to increase within the venous erythrocytes. And once the osmotic pressure increases, what will happen is it will lead to water diffusion into the cell to equalize the pressure and thereby making it large. So answer is option B, the increased osmotic pressure in plasma. So the answer is B or alternative 2. The next one. Which eukaryotic cellular organelles are believed to have come from symbiotic bacteria? So they are mitochondria and chloroplast. So it is endosymbiotic theory. The next one. What is the advantage of having two lipid bilayers around mitochondria? Now the two lipid bilayers separate the mitochondrial content from the cytoplasm. We refer to them as inner and outer mitochondrial membrane and the space between these two membranes is called as intermembrane space and it has a low pH because the electron transport chain embedded in the inner membrane pumps protons into it. And energy to make ATP comes from protons moving back into the matrix down their concentration gradient from the inner membrane space. So the answer is they maintain a proton gradient. They prevent entry of chemicals into mitochondria. No. They act as store of phospholipids. No. So they maintain a proton gradient is more correct. So answer is alternative 3. The next one. The KD KDEL sequence found on the luminal proteins of the endoplasmic reticulum is responsible for. So the KDL sequence prevents a protein from being secreted from the endoplasmic reticulum and facilitates its return if it is accidentally exported out of the endoplasmic reticulum. A protein with a functional KDEL motif will be retrieved from the Golgi apparatus by retrograde transport to the endoplasmic reticulum. So KDL sequence marks the protein for entry into the from being secreted out from the endoplasmic reticulum, and also it will make the protein to retrograde its transport to the endoplasmic reticulum if by chance it moves to the Golgi apparatus. So answer is retrieval of endoplasmic luminal proteins from the Golgi. Then next one, immunoglobulins are classified on the basis of heavy chains. Though this is also a repeated question. In the immune system, the mononuclear phagocyte system comprises of. So the mononuclear phagocyte system has been defined as a family of cells comprising bone marrow progenitors, blood monocytes and tissue macrophages. So the answer is blood monocytes, liver Kupffer cells and kidney mesangial cells. The liver Kupffer cells and kidney mesangial cells are macrophages found in liver and kidney respectively. Then next one, reduced glutathione functions in RBC2. So reduced glutathione are able to protect cells by destroying the H2O2 and hydroxyl free radical. So reduce oxidizing agents such as H2O2 is the answer. Then viruses that possesses reverse transcriptase enzyme and capable of synthesizing DNA from RNA are termed as retroviruses. Which one of the following microbe is linked with Lyme disease? So it is Borrelia burgdorferi. This is also a repeated question. Then next one, which pathway is correct for catabolism of purines to form uric acid? So it is adenylate, inosinate, hypoxanthine, xanthine and then uric acid. So answer is option 3 or alternative 3. Then next one, in the dark, rods show a large inward dark current which is suppressed by a flash of light. Which one of the following statements explaining the effect of light is true? So out of these, the third option, that is sodium channel in the outer segment of rods are closed. That is true. Then next one, which of the following best explains why the plasma membrane of cells exhibit negative resting potential? So the membrane is mostly permeable to K positive and the K positive gradient favors its diffusion into the cell. So that is the answer why the plasma membrane of a cell exhibit a negative resting potential. So answer is option B or alternative 2. Then which of the following replacement causes sickle cell anemia? So glutamic acid at beta 6 position in the beta chain at 6th position is replaced by valine. So GLU beta 6 to valine is the correct answer. Then next one, in contrast to eukaryotic mRNA, prokaryotic mRNA it can be polycistronic. So eukaryotic mRNA are monocistronic and prokaryotic mRNAs are polycistronic. Then in innate immunity, immune cells recognize invading pathogen based on their specific pathogen associated molecular patterns. That is PAMPs through. This is done through pattern recognition receptors or PRR. Then degeneracy of genetic code implies that the genetic code is said to be degenerate because more than one codon can code for the same amino acid. So answer is option D. That is multiple codons must decode the same amino acid. Then next one. Which of the following inhibitor uncouples electron transport and oxidative phosphorylation? So uncoupler of electron transport they have asked, then that is out of these options it is dinitrophenol. In contrast to chemically induced mutation, mutation induced by transposons, they are jumping genes. They keep moving from one place to another on the chromosome. So are more likely to be. So they can revert to wild type very fast because they keep on moving from one place to another. So that is the difference between chemically induced mutations and mutations induced by transposons. So coming to the next question, what is the natural function of restriction enzymes? So they protect the bacteria by cleaving the DNA of infecting viruses. So what does these restriction enzymes they do in bacteria? So when a bacteria is infected by a virus, the virus injects its DNA into the bacteria and this viral DNA is cleaved by bacterial restriction endonucleases, thereby protecting bacteria from viruses. So protecting bacteria by cleaving the DNA of infecting viruses. So answer is option 2. Then bouquet stage in meiosis is seen at. So during the leptotin stage of meiosis, the telomeres of all the chromosomes converge towards the nuclear membrane and assume the shape of a bouquet. So hence leptotin stage is called as bouquet stage. So answer is option 3, leptotin, or option C. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.